Welcome back, everyone. Next, we have Shuttle Pharmaceuticals Holdings, Inc. It trades on the NASDAQ under the symbol SHPH. It's a clinical stage pharmaceutical company leveraging proprietary technology to develop novel therapies designed to cure cancers. The company's goal is to extend the benefits of cancer treatments by leveraging insights into the pillars of cancer therapy: surgery, radiation therapy, chemotherapy, and immunotherapy. Please welcome its CEO, Dr. Anatoly Dritschilo. Nice to see you, Doctor. How are you today?
Good morning, and I'm glad to work with you today.
All right, the floor is yours, Doctor, and when you're ready for questions, call me back and we'll take questions.
Okay, so Shuttle Pharmaceuticals is a company that was established about 10 years ago by three scientists. Myself, I'm a clinician scientist, I'm a radiation oncologist, Dr. Milton Brown, who's a medicinal chemist, and Dr. Mary Young, who is a radiation biologist. And the issue that we want to address is how to improve the treatment of cancer in combination with radiation therapy. The unique aspect of our company is that everything that we do is related to improving radiation therapy for the treatment of cancer. So let me go this. This is our disclaimer to that I'm required to show, and we will go forward and address the fundamental problem that we are facing and Shuttle Pharmaceuticals is trying to address with our products. Basically, radiation therapy is delivered using high cost, very intensive treatment machines, such as this linear accelerator that is shown here, where high energy radiation is delivered to various parts of the body. And this cross-sectional area here, this CT scan with the superimposed doses, is shown to demonstrate what we actually try and do with the treatment. And what we do is use combinations of fields from different angles to get a dose of radiation that's very high to the tumor, but low to the normal tissues, and we try and miss the normal tissues. And as you can see here, this red area, it demonstrates the highest dose that we can achieve. If we go higher than some of these doses that the normal tissues can tolerate, we end up with damage to the patient. That's called radiation necrosis. And the description is really provided by these two sigmoid curves, where the tumor control has a very steep control, meaning that once you get to a certain dose, you start curing patients, and then you get to a maximum dose and it plateaus. So there's just so much you can achieve without damaging the patient. And the complications are related to the dose also, with a small window for treatment, a therapeutic window. The main goal is to make the cancers in here see more radiation than the normal tissues outside. So we have a two-way strategy to address this. One is to directly sensitize the cancer cells to radiation, and with that to improve the control by giving more effective radiation to the tumor, to the cancer, than to the normal tissues. The second way is that after the cells in the cancer are damaged with radiation, they go through a process where certain antigens are activated and the surface of the cell demonstrates these antigens. And this activates the immune system. This particular aspect of the immune system has not been well investigated in radiation therapy. There are some data that show that in some patients this is a very important aspect of the treatment, and others less so. But we'd like to address this by using drugs that stimulate the immune system after radiation in order to help to reduce the cancer cells that are left that may not be completely killed by the radiation. The clinical pipeline for Shuttle Pharmaceuticals is composed of the DNA-mediated radiation sensitization, which are the drugs that are incorporated into the DNA and make the DNA more sensitive to radiation damage. And then the HDAC inhibitor platforms, which are the ones that change the gene expression and the immune response and are able to affect an increase in cell killing by activating some of the body's defense systems. These HDAC inhibitors, these are histone deacetylase inhibitors, are an area that we as a company spent the first 10 years of our existence in actually manufacturing, designing, inventing, and patenting. We have more than 22 patents on various HDAC inhibitors, and we focused on these specific three for development further with the class 2 HDAC6 inhibitor SP2-225, which is the one that we see as activating the immune system. The mechanisms underlying treatment, the clinical lead drug, ropidoxyuridine. This is a drug that has the chemical name of IPDR and is a prodrug of IUDR, iododeoxyuridine. The drug itself has been shown in the past in human studies, the IUDR, of having a positive effect on sensitizing brain tumors and sarcomas. And the data has been published. The downside of the IUDR was that it was not deliverable by oral route. You had to do infusions, and there were some difficulties with that. And so that ropidoxyuridine was developed for an oral preparation where you can take a pill every day before treatment and then get treated and have the effect of IUDR. And the ropidoxyuridine is transformed in the body into IUDR. The IUDR then enters the DNA, and when radiation is applied, the DNA is more sensitive to breaks due to an increase in what's called free radical formation. So this is a mechanism that's well established, and IUDR is recognized as the most effective sensitizer, chemical sensitizer that has been studied. So this is one of those very effective drugs. It's just a matter of how can you deliver it and how can you make it effective for treating cancers. The phase 1 clinical trial was completed with funding from the National Cancer Institute to Shuttle Pharmaceuticals, and in conjunction with collaborations with the Brown University Rhode Island Hospital, we completed a phase 1 trial showing that the drug can be given to patients in combination with radiation and did not show any adverse events that would prevent it from going forward. 18 patients were treated, and a maximum tolerated dose of 1,200 milligrams was established for the phase 2 clinical trial. Out of the 14 patients that were accessible for tumor responses, we saw 4 partial responses, 9 stable disease, and 1 patient that progressed. The recent activities of this, we've taken the information from the phase 1 clinical trial to prepare an IND, and we have met with the FDA and received response from the FDA in a pre-IND meeting. The IND has been prepared using the guidance from the FDA, and the company has also completed all of the necessary manufacturing of the active pharmaceutical ingredient, as well as encapsulation and formulation of the material. So this has been completed. We have also strengthened our technology with additional intellectual property, which includes a patent that was awarded for combining the ropidoxyuridine with another drug called the for a method of use that gives us a higher uptake of the drug at the same concentration. So this improves the bioavailability of the drug. We have expanded our lab space to facilitate further development of ropidoxyuridine's formulations in order to address various other clinical sites for radiation sensitization. The primary intellectual property for this rests initially with orphan designation for the treatment of brain tumors, glioblastoma, so that by using these additional developments, we can then expand our market to include other cancer types. The second area that I mentioned was the HDAC inhibitor platform. We discovered that by using something called the ATM activation site, or the HDAC selective domain. ATM is ataxia-telangiectasia mutated, and it is a gene that has implications for radiation response. And patients, children who are born with mutations in the ATM gene have very strong sensitivity to radiation. So we combine this to activate that particular part of the molecule. So we have this combination molecule, and we use this platform to invent a number of variants, as I have shown you before. The HDAC inhibitors have two functions. One function is to change the gene expression by affecting the DNA by unwinding it and causing it to express genes that are involved in various cellular functions, including cell growth and immune response. In addition, the acetylation by the HDACs of proteins that are involved in various types of responses, including immune response, are also affected. So we have molecules that cover all of these areas. Both class 1 and class 2 molecules are available, and we will be seeking to develop some of these as we advance. This is a summary slide of our intellectual property portfolio. So we have the dual function molecules for HDAC inhibition, a number of US and Canadian patents. We have a number of European patents. And then we have the methods of the combination treatment with ropideoxyuridine and thymidine phosphorylase inhibitors, as well as a number of selective HDAC6 and HDAC and class 1 HDAC inhibitors, which are listed here, as you can see in the slide. The clinical development of the radiation sensitizers at this time is to improve our, is to complete our phase 2 clinical trial of brain tumors. We want to capitalize on the fact that we have an oral preparation. We want to capitalize on the fact that there is only one radiation sensitizer drug that has been directly approved specifically for the indication of radiation sensitizer. Most other drugs are used for other indications in chemotherapy, and radiation is a side effect of those drugs. So we have, let's say, a clear need in this area for direct radiation sensitizers. The ability to then file for, since we have orphan designation, the ability then to file for additional breakthrough designation would be available to us, and we intend to maximize our interest in all of these areas. The second priority is to develop the HDAC6 inhibitor initially, and this is for to enhance the immune activation of cancers after treated with radiation. We also have other class 1 HDAC inhibitors that have effects on breast cancer and on prostate cancer cells, and we will be exploring those and looking for collaborations with other institutions for the further development. The financial overview shows we have about a little over 18 million shares out. The market has been difficult recently. Our stock has been in this area, in this range for the past year, and our market cap is shown here with cash balance. It allows us to advance. We will be seeking additional, funding for some of the development work of the drugs that I mentioned, especially the HDAC6 inhibitors, and we will be collaborating with, looking for collaborators and other, companies that maybe have interest in pursuing some of these patented drugs that we have developed. So that's the 30,000-foot overview of what's happening in the company. I'd like to show you our leadership team that is doing most of this work. And as I've said, I've worked in academic radiation oncology. I'm a physician. I've taken care of thousands of patients with cancer and treated them with radiation. I have experience with industry, and I've served as a director of a cancer center and a department chairman for many years at Georgetown University. Peter is my son. He's an administrator, hospital administrator, and he provides us with administration. Michael Vanderhook was one of my collaborators and workers as the administrative director at the Lombardi Comprehensive Cancer Center, and he has joined us about 4 years ago and has helped us to develop this company. We have a scientific team who includes Dr. Tyven Rich, who serves as a medical director. He trained at Harvard Medical School and the Joint Center for Radiation Therapy, worked at MD Anderson, and has been chairman at the University of Virginia. He has a tremendous experience in radiation sensitizers and many publications. Dr. Mary Young does our laboratory work and certainly is a molecular biologist of very high caliber. And Scott Grunrod works full-time directing our laboratory and is a medicinal chemist and has helped design and patent the various HDAC inhibitors. So thank you very much for listening to my update of the company. And I'd like to go back to Anna. Anna, are you available?
Yes, Doctor. Thank you so much. Let's talk a little bit about your differentiating factor. There are a lot of companies out there developing cancer treatments. So what sets Shuttle Pharma apart from the rest?
Right. So our focus is almost entirely on developing drugs that improve the effects and the outcomes of radiation therapy. So these drugs have to be used in combination with radiation, either during the time of treatment or after completion of treatment. And to my knowledge, I think we're unique in that area. This is our primary focus.
That makes sense.
And I should say to you that radiation therapy is a very important component of cancer treatment, since more than half the patients that are diagnosed with cancer are treated with radiation. That's about 800,000 patients a year that are treated with the radiation at some point in their cancer treatment. So the market is very large, and the drugs that we're looking at have multiple areas that need to be investigated where we can help patients and get better outcomes.
Absolutely. Talk a little bit about your origin story. How did Shuttle Pharma develop, and what's your role in the development of the company?
Right. So, I served, as I said, as a clinician and a scientist, what we call translational research all my life, for about 40 years at Georgetown University after my training at the Harvard Medical School. So the focus has been to develop new and improved treatments. In terms of you take care of patients, but you see where there are needs and you need to address those. And at Georgetown University, we built a team that was working on these areas. And I think that one of let's say, gaps in this whole process is taking something that's developed and advancing it into the clinic and advancing it. You can do that to a certain extent in university. But then once you go to commercialize something and make it readily available to other people, you have to develop a company. So the three of us, as I said, a clinician, a chemist, and a molecular biologist, we formed this company to do precisely that. And we've been doing this for 10 years now.
Fantastic. I also understand that Shuttle has a very rich patent portfolio you've been developing over the years. So describe some of those developments and why they are so important.
So the areas that we focused on primarily is really the HDAC inhibitors. These drugs target the gene expression in cancer cells and in normal cells. So they have a very unique effect with radiation. They sensitize cancer cells and they protect normal cells. And the mechanism is not entirely clear, and there are many people investigating these, but you do see improvement in treatment by having that type of combination. So we designed these drugs and screen them, and we have these various selective HDAC inhibitors. Since there are 12 enzymes involved in the process, there are conceivably at least 12 different targets. And so we've identified some of these targets and we've made molecules specific for that. And all of our work over the last 10 years is coming to fruition and with awarded patents along the way.
Talk a little bit about the size of the market. What other products are available to treat what you mentioned, the glioblastomas and other cancers?
So the glioblastomas, the only other drug that is used with any effectiveness is something called temozolomide. And temozolomide in brain tumors, the glioblastomas, provides a prolongation of survival of 3.9 months. That's less than 4 months. And that's considered to be a big, a home run drug. Now, the target for us is to improve that considerably, and we need to prolong survival in patients. We'd like to cure some of these cancers. So there's a specific subset of these cancers that are extremely, extremely difficult. They're called methylation negative, IDH wild type. And these patients, most of them do not survive 12 months. So what we're looking for is the worst group of patients to see if we can improve the radiation effects in those patients. And so that is our overall goal with the ropiduxuridine. Now, the other drugs, the ones that we will give after treatment, are HDAC6 selective inhibitor. That particular drug is capable of sensitizing after the fact by stimulating the innate immune system. And for that, we have been testing several disease sites, including we're looking at breast and we're looking at head and neck cancer cells. And this has been preclinical, and we'd like to advance that to the clinic.
And what can the sensitizer drugs contribute to the cancer treatment with the radiation therapy?
So the problem with, let's say, just using radiation therapy is that you're limited by the surrounding normal tissue, meaning that if you can give a high enough dose to a cancer, you can cure any cancer. But you're limited if you go too high, you end up damaging normal structures, so that becomes a problem. And by damaging normal structures, you have radiation complications, and you can't go above certain doses because it would be unethical to do it. So if for example, you have a tumor that is inside the brain, and the brain can tolerate only 6 weeks of radiation therapy, that's all you can do. If you give a sensitizer that affects just the cancer cells, you then make the radiation effects a little more in the cancer cells than the normal tissues. So you get the effect of going to a higher dose without damaging the normal tissues.
And so how does the Shuttle Pharma's pipeline address this specific clinical need?
So we have the two approaches. One is to have the DNA, the drug going into the DNA, making the DNA more sensitive in the cancer cells, because cancer cells are more rapidly growing. They take up this drug more, and by giving the radiation, we can kill more cancer cells. And then by using the post-treatment and innate immune system stimulation, we can also add to most other cancer types by looking at improving the outcomes after radiation therapy by keeping the immune system stimulated. Initially, with DNA, with radiation damage, you get many antigens and molecules on the surface of these cells that are not normally there. And that stimulates the immune system. But after a few days, that immune response, which is an inflammatory type of protective process in the humans, that immune system wanes, and then you have what's called a healing phase, where you end up with cells that become stimulated to grow. And there's factors that allow growth. And that actually enhances cancer cells also. So by prolonging the inflammatory phase, we're able to, let's say, prolong the control of tumors and enhance the control of tumors using our drugs.
Really important work you're doing here, Doctor. Talk a little bit about the future. What is next? Next steps, some milestones, and what is needed to succeed for Shuttle?
Okay, so the next step is to take each of our drugs to the next level. The ropidoxuridine, we need to do the phase 2 clinical trial, which we intend to start in a month or two, and then we would then take the phase 2 clinical trial, and showing any efficacy, we would then use that information to guide us with the FDA to definitive clinical trial, phase 3, and commercialization. Because we're dealing with an orphan indication, we may have some advantages with that in terms of depending on how good the phase 2 data is. On the other area, the HDAC inhibitors, next step is to manufacture additional quantities, do the IND enabling studies, and to start the phase 1 clinical trials in specific diseases such as breast cancer and head and neck cancer and lung cancer with those drugs. We have a very rich portfolio there. We don't have enough bandwidth at this time to do everything, so we will focus on the two areas, the two drugs that we're particularly interested with, the HDAC inhibitors, and the others we will be looking for partners that may be interested in working with us.
Well, thank you so much, Doctor, for your time and fantastic presentation today. We would love to have you on in 2024 with some updates.
I look forward to it. Thank you.
All right. Thank you so much. And thank you all for watching. We'll be right back.